Hello, everyone. We appreciate you taking the time to join us today. Microbial Insights is delighted to have the amazing Dr. Michael Hyman, who's going to be presenting for us about the 4 Bs, and this title is just amazing, the basic bugology of biodegradation and bioremediation. Many of you have heard Mike speak for us before, and you all know he really doesn't need any introduction. He's really an amazing speaker, and Mike does such a great job of taking complex material and making it into something that I can really understand and relate to. So I'd truly love to hear him speak. For those of you that haven't heard him before, you're in for a treat today. Dr. Michael Hyman is professor and director of graduate programs for microbiology at North Carolina State University. Professor Hyman is a passionate researcher and speaker who does some phenomenal research on enzymology, pathways, and the physiology of microbes that degrade a variety of environmental contaminants. His group works to understand the mechanisms that are utilized by naturally occurring microorganisms to degrade contaminants, and then to help develop treatment systems to be able to maximize activities in contaminated environments. Now, for those of you that have attended before, you know that this webinar is being recorded and it's going to be available in the resource section of the Microbial Insights website within the next week. A link will also be emailed to everyone who registers for the webinar, so there's no need to reach out to us. You're automatically going to get a link with your certificate of attendance. And after the presentation, we're going to have a short question and answer session. So feel free to go ahead and type your questions throughout the presentation, and then I will read those to Mike, and we'll get through just as many as we possibly can at the end. Now, please note, we do have a very large audience today, one of the largest that we've had. So please be patient if your question we don't get to it immediately. We'll try to get to it, or we'll send out a list at the end with all the answered questions for whatever we can't get to. But without any further ado, I'm going to go ahead and turn it over to you, Mike.
Okay, great. Thanks, Dora. Thanks for that glowing introduction. I hope I live up to it. And thank you all for attending today. This is, as the name might imply, a very sort of low-level description of the microbiology, otherwise known as bugology, that's relevant to biodegradation and bioremediation. And Dora asked me to do this as there was a previous version of this done many years ago, and she felt that this was a good opportunity to sort of revisit some of these issues and sort of represent this material. So as I indicated in the blurb for this talk, you know, I've done several presentations, webinars for Microbial Insights previously, and I would describe some of those as definitely in the weeds. So hopefully with this sort of image here, you can get the idea that what we're really going to talk about is above the weeds. We're not going to get into huge technical detail or perhaps you know, unnecessary detail. We're going to focus on the big issues. And the topics are going to talk about, give you an introduction to the types, roles, and numbers of microorganisms that you might find in environments. We're going to focus a lot on soils and groundwater. We're going to have a very quick summary of the key biomolecules in bacteria, because these are the things that we use a lot to identify and quantify microorganisms, specifically bacteria in the environment. A quick summary of bacterial metabolism, dealing with electron donors and electron acceptors. And then we're going to shift into a discussion about how we quantify bacteria and how we can quantify some of their activities. And towards the end, I mean, this is obviously a short presentation. We're going to very briefly touch on modern omic methods, which are transforming some of the analyses that we discussed above there. And then as a sort of final indulgence, took a few final thoughts to sort of look in the crystal ball and see where this whole field is going, and if we have time to answer some questions. So let's start at the very beginning and look at what are microorganisms. I've listed 4 major types of microorganism here: bacteria, archaea, fungi, and viruses. I will tell you that the vast majority of this talk is going to focus on bacteria. But just starting with sort of a general description, bacteria are universally distributed, they're single-celled, and their defining feature is that they have a nuclear region, like a nucleus, well, that they don't have one. In fact, that there is no membrane surrounded nucleus in these cells. So archaea are very similar in some ways to bacteria in the sense that they are another form of these prokaryotes. They have no membrane-bound nuclear region. They are very, very important. We'll talk about them very briefly on a couple of occasions, but recognizing the name that they're a very ancient form of life, and they tend to be present in extreme environments, very hot, very cold, etc. When we deal with fungi, we're dealing with organisms that can either be single-celled or multicellular, and they do have a true nucleus, so they are eukaryotes. They're known as eukaryotes, and in many ways much, much closer to us in terms of their organization and structure. The last group then are viruses, and I put there a quotation which came from one of the Nobel Prize winners who's looked at, viral structure and function, calling them a bit of bad news wrapped up in protein. They are non-cellular, non-living, parasitic organisms that require living cells to replicate. So what's the environmental role of these organisms? Certainly bacteria and fungi and archaea, if we were to include them, they live and grow by degrading primarily organic compounds. Some organisms will degrade inorganic compounds, and they act as primary consumers. If you have a look in this figure over here, they initiate the degradation of compounds and initiate the cycles of nutrients, including carbon, nitrogen, phosphorus, and sulfur. One of the reasons why we're particularly interested in bacteria is that they have a huge metabolic diversity. And I've put these items in red because these are terms that I'm going to come back and touch upon and define for you later on if you're not particularly comfortable with them. But by metabolic diversity, it means that they have the ability to degrade an enormous number of different types of compounds. And as we'll see, they can operate, well, some can operate in the presence of oxygen, some require oxygen, some do not require oxygen. Those that require oxygen are called aerobes, those that don't are called anaerobes. Fungi are particularly good at degrading biopolymers, things like wood, but compared to bacteria, they have a fairly modest metabolic diversity. And while not all aerobes, they are frequently require oxygen. And the role of these organisms, bacteria and fungi in particular, is to initiate a food web. So they become food for higher organisms like protozoa, nematodes, and so forth, all the way through to mammals and birds, etc. The last point that I want to make in there, which does not appear on that slide, is that viruses, while they're not living, they have an enormous impact on microbial populations. Because they are parasites, they can control the size and the composition and the function of microbial communities. All right, let's move my next slide along here. So if we have a look at the numbers of microorganisms in soil, there's a color-coded bar graph there. This is per gram of soil, a rich sort of organic soil. Viruses, you may have as many as 1 billion microbial, viral particles per gram of soil. To a first approximation, an order of magnitude less bacteria, a couple of orders of magnitude less fungi, which we see here, and then algae, protists, and nematodes. And this trend sort of focuses, the trophic levels, that one thing is eating another, etc. 10 to the 8 microorganisms per gram of soil is a large number. It's kind of difficult to sort of imagine mentally. So I did the calculation, and it worked out that if you have 150 pounds of sugar, so we've got 3 50-pound bags of sugar, you've got roughly 1 times 10 to the 8 grains of sugar in those 50-pound bags. So very substantial number. And obviously part of this has to do with the fact that these are very, very small organisms. And if you have a look down here, a typical prokaryote cell, typical bacterium, is about 1 micron in length. That's a very useful rough guide for the size of these things. When we look at the structural features of bacteria, this is, again, very, very rudimentary, but I'm going to start from the outside. This is our bacterium. This yellow layer is called a capsule. It's an extracellular protective polymer. Can help bacteria attach to surfaces and protect them from materials that are in the extracellular medium. There is a cell wall, which immediately follows, and that's a polymer. We don't really need to know exactly what, but it provides support and shape for the cell in general. And then as we sort of travel further and further inside, we get to the cell membrane. And the cell membrane is a phospholipid layer that effectively separates the living material on the inside from the outside world. Very, very important structure, controls the inputs and outputs into the cell. The cytoplasm is the site of most biochemical activity, and it's really just a big soup of organics, enzymes, ions, water, etc. This is where the underlying biochemistry of the cell operates. Then there is this nucleoid region, which is not membrane limited. That's what makes it a prokaryote. And simple way to look at this is that this is a DNA containing hard drive for the cell. All of the information that is needed to make that cell operate, that bacterium operate, is encoded on this permanently on this hard drive. And the reason I use the hard drive idea is that there are also little independent pieces of DNA called plasmids, and maybe the way to look at these is that these are apps. These are small additional pieces of DNA that provide information that extends the capabilities of the cell. These green dots are ribosomes. These are effectively the sites of protein synthesis. We'll talk about that a little bit later on. And then the flagellum, this is a very, very short, tufty thing. Often these things are many times the length of the bacterial cell. This is kind of the rotary protein outboard motor. This is how microorganisms orient themselves and move around. So if we have a look at some of those molecules, the reason why we need to look at these molecules is that they are going to become things that we analyze and look at and can gain great information from. So we'll start with the phospholipids. Phospholipids are the main component of the bacterial membrane. One way that you can look at this is that they're kind of balloon-like. So it's a thin lipid bilayer which contains the cellular materials on the inside and separates from the outer world. Lipids in general are a very broad group of water-insoluble inorganics, but phospholipids are a particular type of subdivision of these. This is the structure of them. They have a charged head which contains phosphate. Oh, hang on, I just missed my... contains phosphate, which gives it a charge and enables it to interact with water. And then you have these fatty acid tails, which are hydrophobic and avoid water. And consequently, we can have this self-assembling material, which provides this protective and selective membrane from the inside and the outside. The significance of this, obviously, is that if that membrane is ruptured, the bacterium will die. That gives you some idea of its significance. And if you add up all of those, the abundance of these molecules in a bacterial cell, it's usually the most abundant biomolecule present in there. If we move on, protein and enzymes. Proteins are folded, stabilized polymers of amino acids. We have an amino acid there, a generic amino acid there over on the left. They're joined together by peptide bonds. There is a huge sequence variability in these polymers. These proteins, there are 20 main amino acids that get used by biological systems. There are many, many more, but there's 20 canonical amino acids. And there is, again, another reason for the huge sequence variability is that the average protein, if there is such a thing, is about 35,000 molecular weight, which is the equivalent of about 300 amino acids joined into a chain. There are steps of organization in proteins that follow this linear chain, and you can see on the right-hand side there, this is an example of a very non-specific enzyme. This is methane monooxygenase. This is an example of a protein that has a catalytic activity and is therefore an enzyme, whereas other proteins lack catalytic activity and they serve structural roles or functions such as flagella, which we talked about. DNA and RNA are the last things we're going to talk about. These are nucleic acids. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid, and they are both polymers, but they have some very, very significant differences. One of them is the sugar that is in the monomer of these materials. Over on the left-hand side, we've got a deoxyribonucleotide, which goes together to form DNA, and you can see that there is a base, then a sugar, the cyclic structure, and then several phosphate groups. If you look over to the right-hand side, you will see that this is the equivalent structure for the RNA monomers, and the principal difference there is that there is a hydroxyl group, OH, versus a hydrogen group highlighted. There are other differences. There are changes in the bases. If you have a look, there's on the center, there's thymine, cytosine, adenine, and guanine are present in DNA, and largely the same in RNA, except that thymine is replaced by uracil. DNA is typically double-stranded. It is stabilized by having two chains that run in opposite direction with complementary information, which is the sequence of those bases. RNA tends to be shorter in length. It tends to be less stable. The principal organization of DNA is in genes. So these are sort of like the little programs that get run off the hard drive. And RNA, as you'll see a bit later, has got multiple functions, principally in protein synthesis. The central dogma of biology, I know it's sort of perhaps getting a little bit close to the weeds here, but it is very important to understand this flow of information that occurs in biological systems. There is different languages used in different molecules. DNA, as we pointed out, uses A, T, C, and G. That language is different for RNA. And the principal issue is that the information that is in DNA effectively codes the sequence of amino acids in a protein, and there is this intermediary step of having mRNA or RNA in between, which is what the figure demonstrates up there. There are two key processes. Transcription is where you're converting DNA into this other language, into RNA, and the product of that process is called messenger RNA. And then there is the translation step where the information in mRNA is read and then used to direct the polymerization of amino acids by a ribosome. And a ribosome, a picture of a model of the ribosome, is over there on the right-hand side. Key issue then, the take home, is that there is a flow of information in biological systems that goes from DNA to RNA and then eventually to protein. The purists, if you go back and look at the original papers describing this central dogma, it actually says that protein is a dead end. In other words, you can't get back to DNA and mRNA from protein. There is no biological conversion step. But for most purposes, it is recognized that the flow of information, certainly for everything that we're interested in, goes from DNA to RNA to protein. So we've talked about the basic molecules. Let's have a look how microorganisms work. For those of you who are a little perhaps rusty on your chemistry, there is, we're talking about here redox reactions. And there are some terms there which are important. We have electron donors and electron acceptors. And when you remove an electron from an electron donor, it becomes oxidized. And when you give an electron to an electron acceptor, it becomes reduced. And effectively, that is what microorganisms do. They catalyze redox reactions, and there is an energy change which occurs when that electron is donated from a donor to an acceptor, and that energy change is what powers life. In the simplest of terms, that's what's going. It's obviously more complex, and we're going to have a very brief look at how that works in the next slide. Here's a model of a bacterium. You'll see a term there. There's an electron donor over on the left-hand side, and then there are electron acceptors at the bottom there. An electron donor will be taken up by a cell. It will be degraded by what are called catabolic enzymes. When that process occurs, electrons are removed from the original electron donor, and those electrons are then passed to an electron transport chain, which is designated here. And there are a series of intermediates and metabolites which get generated. If you follow the carbon flow, that electron donor, some of that carbon can end up as CO2, so it's been mineralized, or some of those smaller intermediate metabolites can then be utilized by what are called anabolic enzymes to rebuild proteins, lipids, nucleic acids, etc. And you'll see the energy that powers that process is ATP. ATP stands for adenosine triphosphate. Best way to look at it is it's the cash energy currency of the cell. And it's the electron transport process that enables that ATP to be formed. Once we have extracted all of the energy out of those electrons, primarily by making what is called a proton gradient, those electrons get dumped onto an electron acceptor, which then becomes reduced. A couple of points there then, that metabolism really represents the sum of both catabolism and anabolism. And if you find that difficult to follow, there is a simple sort of mnemonic, the ABC of metabolism, that anabolism builds. In other words, it's producing new molecules, whereas catabolism degrades. Simple way of remembering. So let's look quickly at electron donors. Enormous variability. Water, for example, is an electron donor for a lot of photosynthetic bacteria. Those are often called photoautotrophs. The photo telling you where they get the energy from, and the autotroph telling you that they are fixing their own carbon dioxide. Autotroph means self-feeding. And there are quite a few bacteria that do that. Other organisms that do that are organisms that use inorganics like ammonia or hydrogen sulfide. They oxidize those materials and they use the energy to fix carbon dioxide, and they're known as lithoautotrophs, organisms that grow on inorganic materials or stone, litho. And then when we get to organic materials, these materials typically can act as both the electron donor and the carbon source, so they're known as heterotrophs. Some other features that we need to think about electron donors. They can induce expression of dedicated catabolic enzyme pathways. The cell senses the presence of a substrate, expresses the enzymes that enable it to degrade that compound. It's often a misconception that bacteria can only degrade one type of donor or carbon source. That's typically not true. Bacteria can often degrade multiple different types of electron donor, and sometimes many different types of contaminants. We come back to this issue about plasmids. Plasmids often contain genes that encode enzymes for particular pathways. Octane, for example, is the ability to degrade octane is often encoded on a plasmid, and that plasmid can be shared with other organisms. And genes, the genes that encode these enzymes, and genes is a thing that we'll define a bit later, are very, very useful targets for understanding biodegradation processes. When you think about electron donors, most natural environments are carbon limited. So those are called oligotrophic conditions. And the minimum carbon concentration needed to keep a microorganism alive is roughly, to a first approximation, about 1 milligram per liter. And there are key features about donors that don't help their biodegradation. Polymers tend to be difficult to biodegrade. Biology loves nice linear molecules, so branched compounds represent some difficulty. And then the addition of halogens can be another issue. So let's have a quick look at some electron donors and biodegradation. The top line represents some components of gasoline and diesel. We've got hexadecane on the left. That can be degraded both aerobically and anaerobically, the plus of oxygen and minus oxygen. The next one is 2,2,4-trimethylpentane, a major component of gasoline. It is biodegradable in the presence of oxygen, not aware of degradation under anaerobic conditions. And it degrades slower than hexadecane onto its left because it contains branches. We've got an example there of benzene. Benzene can be biodegraded both aerobically and anaerobically. And then the same is true for MTBE. We sort of drop down a bit into the next level. 1,4-dioxane over on the left-hand side is known to be biodegraded and used as an electron donor under aerobic conditions. There is no real evidence or strong evidence that it can be degraded anaerobically. And then when you look at some of the co-contaminants that you find often with 1,4-dioxane, like 1,1,1-trichloroethane and 1,1-dichloroethylene, the next two, 1,1,1-trichloroethane can be used as an electron donor. Dichloroethylene potentially can, but there are problems associated with it, and they are related to the double bond that you see there. And then at the bottom level, there's some examples of things that degrade more slowly. Over on the left-hand side, we've got lignin. Lignin is biodegradable, but it tends to be slowly biodegradable because it's a polymer. The next one there is perchloroethylene, tetrachloroethylene. Does not biodegrade aerobically for sure. And the reason being is, well, one of the contributing factors is that there is no carbon-hydrogen bond in that molecule. And it's the carbon-hydrogen bond which represents a major source of energy when you're degrading these compounds. And then the last one there is a representative of a PFAS type compound, really slowly biodegradable or potentially not biodegradable at all. The issue there is that there are, again, no carbon-hydrogen bonds. They've all been saturated with fluorine. The potential degradation is dictated by what X actually is. There is one other subtle twist to the way organisms can biodegrade compounds, and that is called co-metabolism. And effectively what we're doing in these situations is using a readily biodegradable electron donor, we talk here about methane or propane or isobutane. This leads to the expression of a pathway which enables the microorganisms to grow. That's up at the top. This is the metabolic pathway. And that leads to the expression of monooxygenase enzymes or other types of enzymes, but monooxygenases are a big focal point. And that enables the expression of those enzymes, those monooxygenases, enables these bacteria to degrade things like MTBE, 1,4-D, trichloroethylene, but actually not use them as either carbon or energy sources. So that we're capitalizing on the degradation of one compound to power the degradation of another poorly degradable compound. And it's actually a very, very useful approach for degrading contaminants, for example, at very, very low concentrations, part per billion to part per trillion sort of concentration levels. Switching terms, then let's look at the other end of the spectrum. Then we're dealing with the electron acceptors. There are a series of common electron acceptors here: oxygen, nitrate, iron 3, manganese 4, sulfate, and CO2 going in sequence. The size of the arrow is just nominally to reflect the fact that oxygen is often a preferred electron acceptor because the amount of energy yield that you get using that compound as an electron acceptor is the largest out of the common acceptors that are present. And so nitrate is less effective as an electron acceptor than oxygen, and so forth as the size of the arrow decreases. The consequence of getting less energy as you use different electron acceptors is that the organisms grow more slowly, and that these electron acceptors tend to be used in this descending sequence in contaminated environments. So oxygen will be consumed first, and if all the other electron acceptors have been consumed, eventually it will turn to the production of methane using CO2 as an electron acceptor. It should be clear that there are no organisms that can do all of these, so they tend to be reflect somewhat specialized organisms. Sulfate reduction, for example, is the domain of SRB, a fairly coherent group of organisms called sulfate reducing bacteria. Methanogens are actually archaea, they're not bacteria, but these are the organisms that produce methane. So, and some of these things are a little confusing in the sense that they have different names. So CO2 reduction can be known as methanogenesis, making methane. The reduction of nitrate to N2, for example, is also known as denitrification. But these materials are actually very, very useful because you can monitor the concentrations of these in field conditions and come up with some idea about what actual microbial process might be occurring. So there are test kits for oxygen, nitrate, the presence of iron, for example, concentrations of sulfate, and etc. The key issue that I do want to point out just before we leave this is that if you use oxygen as an electron acceptor, you are defined, the organism is defined as an aerobe. If you use anything but oxygen as an electron acceptor, you're defined as an anaerobe. And lastly, the last point is that I should point out that not all electron acceptors are benign. And following what we did for the electron donors, here are some electron acceptors. Top left, we can see the reduction of oxygen through to water. There are problems sometimes when we get less than complete reduction of oxygen to water. We can produce things like superoxide, hydrogen peroxide, etc., which are potentially damaging materials to a cell. If we move over onto the right-hand side there at the top, we can look at the sequential reduction of PCE to TCE to dichloroethylene to vinyl chloride and ethene. This is obviously a very big issue in the bioremediation industry. In these cases, there are many organisms that can do some of these reactions, very few that can do all of these reactions, but they are being used as electron acceptors for metabolic processes. There are some curious ones. If you look down the bottom left, this is the issue of perchlorate reduction. Perchlorate, another common groundwater contaminant. This is viewed as an anaerobic form of respiration. In other words, it doesn't require oxygen. The kind of irony at the end is that once we have reduced perchlorate through chlorate and chloride, the final step actually produces oxygen, which is a metabolic interest. And then we can also use other materials. They don't have to be organics or inorganics or organics as electron acceptors. The last panel on the bottom right looks at the potential mechanisms for reducing materials like chromium and other metals. Some of these can be used as electron acceptors to support growth, but obviously the ability to remove these metals through biological activity and often involving their reduction or sequestration is a very big part of of microbiology. So let's sort of switch gears and look at quantification. We often want to know how abundant specific microorganisms are, and there are plenty of old school approaches which involve time-consuming activities. So we can use plates. So if you have a look at the bottom left, there's a picture of somebody's hand that they placed on a Petri dish, and like a high school type experiment. And you can see the enormous diversity of different microorganisms that have grown up there, different colony shapes, different colors, different forms, etc. Plates are not a great way to quantify bacteria because they can be very, very selective, and they often use media designed for quick testing of fecal enteric bacteria like E. coli. And on the flip side, microscopy struggles with quantification and the resolution between live and dead and detritus. So perhaps if you want to have a look at that middle image there, good luck telling me which, and counting which are the living and the dead organisms there, which ones are bacteria, which ones are bit of detritus. So a lot of the approaches that now get used focus on culture independent methods, and they look at biomarkers, and these, as that right hand image suggests, are frequently genes which we find in DNA. So before we go much further, we should perhaps define a few of these terms that will get bandied around. We've talked about how nucleotides get polymerized. If we start right in the middle here, this onion sort of effect, nucleotides, ATCG, get polymerized to form nucleic acids. Within the chromosome and plasmids that you have in bacteria, those nucleic acids are organized into genes. A gene defines the information necessary to make an independent protein, whether it be a structural protein or whether it be an enzyme. And if you take all of the forms of DNA within a cell, within this white boundary, that represents the genome of the organism. It's the total DNA that we find in a species. When we sort of look a bit bigger, we can look at what is called the pangenome, and this is the total DNA that you find in all the strains of a species. And so that indicates that there is often considerable variability between organisms which are supposedly the same species but have different capabilities. And then the big red one is the metagenome. This is the total DNA from an environmental sample. All of the organisms that are present in there. It's all of the genetic information that they have. So what information can we get from genomes? If you look down at the bottom there is a genome. Each one of those lines represents a gene for a particular enzyme or other molecule. And there are effectively two different types of analyses that we can do. One of them is to look at the gene that encodes one of the RNA molecules that goes into a ribosome. This molecule acts as a molecular clock. In other words, it undergoes sequence changes that are reflective or at a similar pace to actual microbial evolution. And if you can analyze that sequence, you can start to identify an organism, and you may well be able to infer some of its metabolic capabilities. So the way to look at a 16S ribosomal RNA is that it is the business card of a microorganism. It tells you who it is and gives you some hint about what it does. The other route that you can take is to look for functional genes. So in other words, these are genes that encode proteins that have, which are enzymes. And as you can see, we go through the transcription and the translation process. And the enzymes are the things that we're particularly interested about because these are things that catalyze activity. The way we can analyze that is typically using quantitative or polymerase chain reaction, qPCR. The great thing about PCR is that it is a simple reaction that enables you to amplify a small target piece of DNA within a large assembly of other pieces of DNA. So it's the ability to amplify the needle in the haystack. It's an enzymatic process. It goes through repetitive thermocycles. So the machines that are used to do this are called thermocyclers. The piece of DNA that you want to amplify, the needle, is defined by short primers. And if you go through enough of these cycles, you can generate upwards of, near to 1 billion copies of identical DNA from a small starting material. PCR itself is used generally to amplify DNA. Quantitative PCR provides the mechanism for determining or quantifying how much amplification of your target has occurred. And it works on the idea that the more of a target you have, the faster your amplification will exceed a fluorescence threshold. So we monitor the amplification process by fluorescence. And once that fluorescence exceeds a baseline, you can use that number of cycles to quantify how much of a gene was present in the original sample. There are different variations on this. There's a process called TACMAN, which is by far the more sensitive and reproducible and specific than the general CYBER method, but they both work by looking at fluorescence. So, okay, so that's one of the ways that we measure gene abundance. So we can look at which organisms are present and potentially what enzymes are present. We also often want to quantify microbial activities, and this is somewhat of a deliberate segue in there. There are many non-specific assays that can be conducted for active biomass. So they look at universal biomolecules such as ATP, how much ATP is present in an environmental sample, or looking at ubiquitous enzyme activities, esterases, for example, things that break ester bonds. Every organism has these things. And there are assays that can be designed to estimate the amount of biomass based on these things. But often we're more interested in specific processes. And I've listed a few examples there. I won't go through them in great detail. But jump to the last point that many of these methods for investigating microbial activities, because they tend to involve relatively small molecules, some of them gases, etc., are very amenable to analysis by gas chromatography and in conjunction with mass spectrometry. So we'll have a quick look, refresher on how gas chromatography works and some of the changes that can get used and what they can tell you. So on the left-hand side, we've got a typical gas chromatograph. You have a gas cylinder, which is connected through a column. That column is heated, and the column contains material, a solid phase, which will enable the separation of a complex mixture when it's introduced at the inlet. At the inlet, it's heated, it volatilizes all of the analytes. They get picked up in the stream of the carrier gas, transported through the column, and they undergo chromatographic separation. And we can then determine the separation and the quantification by looking at the chromatogram. The problem with conventional GC is that it effectively relies on co-elution of an unknown with authentic standards, although you can sort of identify compounds a little bit more equivocally using selective detectors. So there's a flame ionization detector, FID, an electron capture detector, very good for halogenated compounds, etc. We can dramatically improve the analytical capabilities of GC by adding in a mass spectrometer instead of using one of those FID, ECD detectors. And because we get ionization of the analytes as they come off the column, we can get much more unequivocal identification of the compounds. And that's why GCMS frequently gets used for standard analyses of contaminants, things like 8260s. And we can improve the limit of detection of both these techniques with sample pre-treatments, which effectively concentrate the analytes before they go onto the GC. One of the most important things that we can do is couple GCMS with isotopes. And if you have a look at the figure on the left, I always find this kind of interesting. On the left hand of the x-axis, you can see the atomic number defined by the number of protons in an element, and the y-axis is the number of neutrons. And basically what that figure shows is that the black line that weaves its way through the center of that represents the number of stable isotopes for a particular element, and the blue and the brown represent the unstable radioactive isotopes. So stable isotopes are a small component. Radioactive isotopes are a big component of what we can find for individual elements. If you have a look up the top on the right-hand side, we've got an example of this: carbon-12, carbon-13, and carbon-14. Carbon is defined by having 6 protons in the nucleus. Carbon-12 has 6 neutrons. Carbon-13 has 7 neutrons, and carbon-14 has 8 neutrons. And so when you add your 6 and 6, you get 12. Hence, we have carbon-12. 6 and 7 gives you 13. Carbon-13. So actually, if you look at carbon, there are actually 15 different isotopes, C8 to C22. 13 of them are radioactive, and you can have a look at the range of half-lives there. It's huge. But only 3 of them are naturally occurring. That's carbon-12, 13, and 14. And they have very, very different natural abundances, which makes them very useful. Carbon-12 is about 99%, carbon-13 is about 1%, and carbon-14 is at very trace levels. And we can use these things for different things, and that's what we'll talk about in the last few minutes here. Other biodegradation, useful stable isotopes: deuterium, H2, N15, etc. Compound-specific isotope analysis is one example of something that can be used to investigate many features of degradation reactions. If you look at the top panel there, effectively what we are now doing is utilizing the separation power of a GC and the mass resolution capabilities of a mass spectrometer, and we are interspacing it with a conversion, a chemical conversion. And for example, if we are interested in looking at carbon, we can convert all of the carbon containing materials into CO2 using a heated oven between the GC and the mass spec. We use this a lot because biological processes tend to discriminate and act slowly when we're looking at bonds that contain C13 or heavier isotopes such as C13 or deuterium. It just requires a little bit more energy to break that bond than a C12 bond, for example, and hence the reaction goes slower. And during biodegradation, then, effectively, the residual substrate in a system gets heavier. So there is more C13 to C, the amount of C13 relative to 12 increases. Conversely, the products get lighter, so there is less C13 to C12. In other words, the C12 compounds get degraded first. You can look at multiple elements, carbon, hydrogen, chlorine, for example, but you can't do them simultaneously, at least currently. They have to be done on different machines. And it's extremely helpful. You can learn about the extent and mechanism of biodegradation, or degradation in general, what the contamination source is, what the contaminant production process were. And often the data is presented as these dual isotope analyses, which you can see on the bottom left, for example, looking at the change in the isotopic composition with respect to hydrogen and carbon. So there is an application of C13 to biodegradation. If you look at the left, we've got compound-specific isotope analysis, otherwise known as CSIA, and that deals with low natural concentrations of C13 in materials. We can use C13 for other analyses. Phospholipid fatty acid stable isotope probing uses a higher abundance, so we are adding in a substrate which contains C13 to a higher concentration than ambient, and we can follow that where that C13 goes, and principally into phospholipids. And that can tell you a very helpful piece of information. Can biodegradation occur? And then there's a more sort of research-oriented version called RNA, DNA stable isotope probing, which actually looks at incorporation of isotopes into nucleic acids. Very, very quickly go through this. The principle behind it is that if you start with an initial microbial community, everybody is largely, all of the biomarkers, all of the biomolecules are labeled with carbon-12, and that the organisms are generating carbon-12, CO2 when they metabolize things. If you then add in a C13 labeled substrate, you're trying to find the organisms that can potentially degrade this or demonstrate that the compound is biodegradable. One organism in this little model has the ability to utilize the C13 labeled substrate. When it does, its biomarkers become C13 labeled, it generates C13 CO2, and its abundance increases as it grows on that compound. And so effectively, that's how SIP works. And one of the most widely used, certainly in the diagnostic side of things, is to look at phospholipid fatty acids. If we go back and reflect on the structure of microbial membranes, remember these are the most abundant biomolecules in cells. And if you assimilate, if a bacterium assimilates a C13 labeled compound at above normal or ambient concentrations, that C13 concentration in these lipids will reflect that. So we can extract and hydrolyze these phospholipids so that we break the bond between the glycerol in the head and the fatty acid tails, and we can then convert them into a GC analyzable material, and we can then analyze them by GCMS or GC-IRMS, an isotope ratio mass spectrometer. The incorporation of C13 into these lipids provides a very, very strong evidence for contaminant biodegradation. You can potentially tell broad groups of microorganisms that might be responsible for this by looking at the different types of fatty acids that become C13 labeled. And that's certainly one of the approaches that can be used if you use biotraps, where you have materials that contain C13 labeled substrates. They're introduced into an environment. Microorganisms grow, native microorganisms grow on these carbon beads, and you can then analyze them for whether or not you've had C13 label incorporation. The DNA or RNA SIPs is effectively a research tool, much more involved, requires much, much higher concentrations of C13 or other stable isotopes. But it does have the advantage that it can identify specific organisms that are degrading compounds rather than broad groups. Lastly, we'll talk about radioactive isotopes. Not the world's most loved form of analyses. They're very, very helpful if you're looking at ex situ microcosm-based degradation processes, because you can add these compounds in at very, very low concentrations, which might be environmentally relevant. And you can then monitor, for example, if you're using carbon substrates, which are common for microbiological assays, looking at the mineralization of these compounds into CO2. And the trick is that you can have very, very sensitive detection using things like liquid scintillation counting. Problems are you cannot do this in the field or realistically, and there are a lot of other things that factor against it. Sometimes you have to get the compounds synthesized and then need to further purify them. That can be expensive. They're radioactive, the compounds, so they have an inherent instability. you have to dedicate particular equipment to the use of radiochemicals, and then you have to get rid of the waste. Despite all of those negatives, these are a couple of papers that have had a very, very important impact on our understanding of the natural attenuation of 1,4-dioxane. I've put the references up there if you're interested. They both make use of a C14 assay. So while there are many, many aspects that make use of radioactive chemicals difficult, they can still be extremely useful. The last sort of slide then is dealing with what are called omics. Omics are a series of very, very data-intensive analytical approaches which are transforming our understanding of microbiology and the processes. If you look at the top, there is a single analysis done for single organisms. We can look at the genome, the genomics of an organism, sequence the genome, find out all the genes that are present. We can look at whether those are expressed, and that's a process looking at mRNA and looking at transcriptomics. We can then see when those proteins that get generated, and that's called proteomics. And then we can also look at what the products of those enzymes are, which is metabolomics. And you can follow the red arrow, and to a very first approximation, these pieces of information move from what could actually happen, in other words, the genes define what an organism might be capable of doing, through to actual measurements of what is happening when we look at what proteins are expressed and what metabolites are being generated. That's all very well and good if you do it for single organisms, but clearly the trend is moving towards looking at mixed communities, and then we have to do these analyses for entire communities, not individual organisms. And then the prefix meta gets placed onto them. So metagenomics is the analysis of DNA from entire microbial communities. Same thing for transcriptomics, same thing for proteomics, so we have metaproteomics. I haven't come across a term for metametabolomics. There's probably one out there. It would look a little silly being metametabolomics, but you understand the principle there. Just to give you some idea of what the issue is, if you look at a single organism like E. coli, the genome itself is 4.5 million base pairs, so the individual nucleotides, so that's a lot. That encodes somewhere in the region of about 4,400 genes. Some of those are RNA molecules, and they do not get further translated, but a very, very large proportion of those are proteins, and many of which are enzymes, and they do get translated, converted into protein. The E. coli metabolome is about 3,000 individual metabolites. If you have an organism with more genes, it means it can do more. That means that you have more metabolites. So imagine the analytical issues you get if you then start looking at your 10 to the 8th bacteria present in the soil, and that you might have several thousand different microbial strains and species present within that community. So analytically, data analytically, very tough. So my last couple of points then, these are my final points, if you can sort of see the crystal ball in the background, sort of somewhat future looking. There are very well validated tools that are available to quickly, accurately, and inexpensively quantify microorganisms and their activities in the environment. Hopefully I've given you a good overview of some of the main ones. But as always, our capabilities and ultimately our knowledge, we can always improve them with technology increases. And the prediction then obviously is that these omic techniques will continue to develop and mature. And while some of the costs are expensive at the moment, you can reasonably expect some of those will fall. The sort of warning point there would be that you probably want to carefully define the question you're asking to make sure that the more data that you get generated by these modern techniques doesn't make the answer harder to find than previously, but that people there to guide you in that. I certainly have a lot of research interest at NC State in microbiomes describing all of the microorganisms within a community and understanding how they interact. And it's very clear that remediation-specific microbiomes can be or are being identified and characterized and ultimately manipulated. And in the end, I think that is going to lead to, just like we have field-specific, literally field-specific precision agriculture, we're going to end up with site-specific precision bioremediation, where we can tailor-make individual organisms or communities to address particular contaminant problems. And then my last slide sort of hopefully summarizes the big picture, and I'd be happy to answer any questions.
Thank you so much, Mike. That was an amazing presentation. We do have a few questions. So if you have additional ones, go ahead and enter them into the question toolbar on the GoToWebinar, and we'll get through as many of them as we can. So Mike, this question is, what do you think about dechlorinating bacteria thriving in high sulfate environments, such as greater than 500 micrograms per liter?
I'm sorry, so could you just go back over again?
Sure. So can dechlorinating bacteria thrive in high sulfate environments, like sulfate greater than 500 micrograms per liter?
I think there are probably better people to answer that's very in the weeds question than myself. But you know, the issue of salinity, I guess the question is, is the person interested in whether or not these organisms can thrive or survive in an environment where there is a lot of sulfate, for example, salinity coming, well, sulfate often associated with salinity, and what is a sulfate reducing environment versus a non-sulfate reducing environment. Salinity, you know, there are specializations that marine organisms have which enable them to thrive and survive in the marine environment. They produce osmoregulatory compounds which are there to prevent water rushing out of the cell and contain water within the cell. But to answer that question specifically, I'd have to have a bit more information, and I must admit that anaerobes are not where I spend most of my time looking. I'm much more in the aerobic world. So I'll answer it as best I can, but dodge the main point there.
No, that's okay. And I can add a little bit to it. We worked on a very large site for almost 15 years now, and we did a lot of statistical analysis. And what we found at this site in particular was for Dihalococoides to be at higher concentrations, we needed the sulfate to be less than 50 milligrams per liter. And so in this site, they had, 100. So we'd have to see them kind of burn through some of that sulfate before we saw active reductive dechlorination. But I have seen that number varies based on the site that we're looking at. So sometimes, sites that have higher sulfate, depending on the communities that are present, can still, Dihalococoides can still thrive and grow. So it is very site dependent. But, once you get below that 50, that was kind of the magic number for that site in Australia. Okay, so sorry, Mike.
No, that's great. Much better answer than mine.
That was good. Okay, so this next two questions are about PCR, and what they're wondering is, when you're talking about, modern PCR techniques, are we trying to identify, first question is the total number of species or the total number of they're saying gene transmittance for genes associated with degradation. And then the second part of the question is, how do we know if these are active microbes? Do we have to follow up with an additional technique to understand that?
Okay. So the first question has to do with specific organisms versus specific enzymes, if I remember correctly.
Yes, that's correct.
Yeah. So you could let's take, for example, methane oxidation, which is an activity of great importance. So these are catalyzed by aerobic organisms to a first approximation. There are other systems that degrade methane. There are many different species of methanotrophs, bacteria that can aerobically oxidize methane. but they tend to have one of two different types of methane oxidizing enzyme. You could potentially develop PCR primers, which would enable you to capture the broad world of methanotrophs, and people certainly do that if you are trying to come up with estimates of diversity, etc. But in most cases, you're not so concerned about an individual organism than you are about how they actually oxidize methane. And so in that particular case, you might emphasize looking at particular enzymes over particular organisms. The flip side is that you have unusual circumstances, for example, like the reductive dechlorination of PCE all the way through to ethene, which is catalyzed by a very, very limited number of microorganisms. And consequently, in that case, it probably makes much, much more sense to have qPCR primers that can quantify that specific organism or very, very closely related organisms, rather than the enzymes that they use to achieve the reductive processes. So it's situation specific. I would say in general, I think that, organism specific PCR, qPCR is probably more the domain of reductive dehalogenation processes than it is for aerobic biodegradation processes. But it really does depend on the question that you're asking. If you are interested in microbial diversity, you want to know how many of each different type of methanotroph is present in an environment. That's a different question than asking how much inherent ability is there within a sample to degrade methane, which you would address by quantifying the number of genes that encode these methane oxidizing enzymes. Does that clarify that question? Was there a second part? I've forgotten what the second part is now.
The second part was about live versus dead cells. Do they have to follow up with an additional technique, or is there a way to do that with PCR?
Difficult. You can work on the idea that, if a dead cell likely occurs through destruction of the membrane, it's not always, but it's a good case. You can certainly discriminate between live and dead cells using stains, microscopy. Less ability to do that with a molecular technique, but you can, to a very first approximation, assume that the half-life of DNA from a cell that has been lysed is going to be considerably shorter than an intact cell. DNA, actually, you can grow bacteria quite well on DNA. They love it. It's got sugars in there. It's got nitrogen in there. So, exogenous DNA can be degraded. And so the live dead is something you need to be aware of. But certainly to a first approximation, I think it would be reasonable to assume that most of the detections you're getting are from living bacteria. It doesn't, or at least living or napping bacteria, because a lot of bacteria spend their time not in a rampantly active state in the environment. They are waiting to be, if there's very little carbon, there's very little they can do. They're just sitting around waiting for a carbon source to arrive so they can leap into action. The question about whether the abundance from a gene-based qPCR assay tells you whether or not the enzyme is being expressed is one of the limitations of qPCR, and that's why I think there is a push towards proteomics, which will enable you to identify an enzyme which has been expressed. It's not just a potential, which is what really qPCR can often provide you. And then ultimately, you're looking for evidence that the cell is alive and actually catalyzing these reactions, and that perhaps leads you into metabolomics. And frankly, some of the metabolomics we kind of already do, in the sense that we look at the transformation of electron acceptors or consumption of electron donors, etc. Metabolomics, I guess, in the purest sense, describes all of the intermediates that are present within a microbial cell or community. When we've already started looking at a subset of that. So certainly proteomic approaches lead you into a much closer to actual catalysis. You know that the enzyme is being expressed. And so if you can quantify the protein, that gives you an added sense of security over just measuring potential through a qPCR assay for a gene.
Okay. The next question, can you talk about the use of nutrients to improve biodegradation, and if there's any recommendations that you have on that?
Yeah, we didn't talk much about nutrients. We focus primarily on electron donors, which are invariably or predominantly organic, and electron acceptors. There are 6 basic elements that need to be supported in the growth of a microorganism. Those are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Organic materials tend to contain a lot of carbon and hydrogen and oxygen. Nitrogen, phosphorus, and sulfur. Out of those, sulfur, the most common source of sulfur for bacteria to grow is sulfate. Not that they use that as an electron acceptor, but they are physically incorporating that sulfur from the sulfate into the biomass. So the 2 big ones are probably nitrogen and phosphorus, and they are, nitrogen is frequently a limiting nutrient. So adding a nutrient mix that contains nitrogen is potentially very helpful. You might want to think about what form that nitrogen is in. Ammonia, if you put ammonia in, you potentially can have that consumed by ammonia oxidizing bacteria. So nitrate can sometimes be a better choice, and there are other options. The other thing that you need to think about in there is that a lot of the enzymes that catalyze some of these key reactions are metalloenzymes. So they incorporate things like iron or molybdenum or cobalt, and these are required at very low concentrations. But if those are missing from the media or the local environment, adding low concentrations of these can potentially stimulate growth. Good example of this, looking at what the impact of nutrients would be the one I always bring up is wonderful pictures from the Exxon Valdez spill, where you're looking at oil-soaked rocks on the beach, and one side of a line has been left to weather, and the other side of the line has been treated with a nitrogen phosphorus fertilizer, and the difference is huge. I mean, the fertilized area has dramatically lower concentrations of residual oil. The place where adding nutrients can be a big deal is when you have got high concentrations of organics, and you are sort of overwhelming the ability of the natural environment to supply the nutrients needed to support that biomass. So for example, if you're using a co-metabolic biostimulation process where you're sticking in methane or propane into the subsurface and stimulating rapid growth of methane or propane oxidizing bacteria, adding in nutrients to support that supplemental growth is invariably a good idea.
All right, we'll do one more question, Mike, and then there are a few others that I'll send out to you, and we'll let Mike answer those, and we'll attach those to all the attendees so you can have it if that's okay with you.
Yeah.
Okay, perfect. And then there were a bunch of comments about how great this presentation was, that people really enjoyed it, and they also would like to have a PDF of the presentation. Is that okay for you to send me?
That's absolutely fine. Absolutely fine.
Okay. Awesome. Okay, here's our last question. So when we're
Okay.
sampling groundwaters from microbial populations, do you think it would be beneficial to also collect saturated soil from the same zone? And if so, could you tell us what that could potentially identify for us?
Ooh, okay. So the way I look at this is that bacteria, microorganisms are very practical organisms. They don't do things for no reason, or the things that they do for very specific reasons. One of the things that bacteria in general, there are clearly cases where it's not so, but in general like to attach to surfaces. And you can see many reasons why they do that. For example, they often attach to surfaces in the form of biofilms. A biofilm can provide protection to a microorganism from predation or antimicrobial compounds or dehydration, etc. So in general, microorganisms, I would say, prefer to be attached to surfaces. Some are prolific attachers and others not so. But when you measure microbial abundance in groundwater, you are measuring the abundance of microorganisms that effectively are not attached. Whereas in soil, you would often be investigating, organisms that are attached. The ratio between the two, I think, is very species dependent. I know Frank Loeffler has addressed this issue specifically looking at dihalococoides and reductive dehalogenating bacteria, dehalorespiring bacteria, because it's much easier to measure. If you don't have to extract microorganisms from soil, it's a more complex process than just analyzing them direct from groundwater. So I would argue probably, if you want a take-home message from that, that the abundance of microorganisms that you are determining in groundwater is a conservative underestimate of the total microbial load within a particular environment if it contains soil or sediment.
Great answer, Mike. And we keep getting
You
comments.
guys have much more experience, I imagine, or can give you specific numbers on the relative abundance, soil versus attached. Part of it's also dictated by how old the organisms are, and that sounds a bit curious, but we certainly have worked on extracting organisms from soil and using differential centrifugation techniques. Process, a thing called Nicodens is what we've used. And certainly in the background research to that, you'll find that organisms that perhaps grow slowly, nitrifying bacteria, for example, bugs that grow on ammonia, are very firmly attached to soil particles. Whereas, and the reason being is that they low activity over protracted periods of time. The same might not be true for a rapidly growing organism that has suddenly been stimulated by the addition of propane or some new novel source. So the ability to detach the organism, or the extent to which it has attached to the surface, may be different depending on basically the age of the culture. But I know you guys you've warned me many times about the dangers of extracting things from soil. So
Yeah, we have to be really careful, and we just don't do that as often as we do groundwater. So we definitely see more variability in the soils that are received, and we have to think about the conditions and the clay content and interferences that can happen, downstream after extraction. So we can do it, and it does happen on a fairly regular basis, but we definitely have a lot more experience with groundwater. And I think we're learning more about the vadosone interactions and looking at soil in those areas. So if you have questions specific to that or a contaminant, feel free to reach out to me. It's something that, I was just talking about in a meeting yesterday, and I think we're going to start seeing more and more comparative studies of those. So happy to share the information. But thank you, Mike. I always enjoy hearing you speak, and that's the response that we're getting is that, I keep getting over and over, amazing presentation. Thank you for simplifying a very complicated subject. And I think that's certainly the fact of what you did today. And so I'm grateful that you took your time and, spent all this time with us. So we really appreciate all the information that you gave us. And, I appreciate everyone taking the time to listen and to join with us. The recording will be available on the Microbial Insights website, so you can always go back and listen to it at any point that you wish. If you have additional questions, feel free to reach out to us at info@microbe.com, and we'll get with Mike and get those answered for you. So thank you again, Mike. I really, really appreciate your help.
Thank you, Dora, and thanks for everybody who turned up. I hope it was helpful and useful.
All right. Thank you, everyone.